cells, which we'll talk about in a second. Produces energy, exactly. And so that energy is called ATP, just so that you guys know. All right, awesome. I think a protein, though, what happens is it's going to make the ATP. And it does have DNA, and I think that might be the confusion. Mitochondria has mitochondrial DNA. So inside there is DNA, and it can make proteins, but the function of the mitochondria is to make ATP. It's to give you the 38 ATP from one glucose molecule. So watch out on that one, guys. All right, perfect. Now, what I want is, okay, ready? Lysosome. Who remembers the little thing? All right, lysosome. Somebody gave me the petite. Yeah, keep moving. Awesome. Good job. Awesome. Sorry, I'm enjoying this. All right, yes. So careful, guys. Lysosome is going to be the purple dot. So not the yellow dot, guys. Move over to the large. So thank you for the large lysosome. And then I want, what does a lysosome do? Tell me what the lysosome does. Does anyone remember what the lysosome does? And remember, these are, I mean, you can do a quick Google search. It's all in your textbook, of course. Destroys toxic stuff. It breaks down things. Because in the lysosome, it's basically just a sac, and it has enzymes. One of the things it does, the lysosome can do some detox and some metabolism, some digestion. But it also breaks down the bacteria. Yeah, it eats bacteria. So that's kind of one of its hallmarks. It's usually used for something like that. Where the lysosome, everyone go to the peroxisome. So peroxisome is not purple. Peroxisome is yellow. So get off the purple and go to yellow. Nobody should be on the purple. Stay away from the purple. Peroxisome. Awesome. Lysol kills bacteria. Oh, I like that. Ooh, Jennifer's got a good one. Lysosome, Lysol kills bacteria. Ooh, nice. That's awesome. Great. So the peroxisome is the petite, the yellow. And what does the peroxisome do? Now, they're very similar, right? Lysosomes and peroxisome are very similar. They used to think that they were like super similar. We often think of peroxisomes being more in terms of breaking down fats. Exactly. So that's where the metabolic kind of piece in that comes into. And yes, it does do detoxification as well if you're thinking about like just toxins in general. And also, guys, all the medication you take, if you take any, or just Tylenol, breaks down in the liver. Okay, so we're going to have to break that down, and that's why we use these. We use the peroxisomes as well. All right, that's great. Awesome. Let's do a couple more.
Question. Yeah, absolutely. What verbiage exactly would you guys accept on the test? Like if we put detoxify,
I'll accept anything that is from the textbook, because some of these have a couple different functions like we've been talking about, right? So for example, we usually think of lysis, the one that kills the bacteria, right? So that's how we would do it. The peroxisome, we think about it in terms of metabolism or breaking down fats. So those are kind of the two big ones, but they do kind of overlap. And as long as you get it from the textbook, then you're good to go, right? That's the main thing. Just make sure you get it from the textbook, and then you can choose one of any of these options. Because again, they do a few different things. Does that make sense, Gloria?
Yep, thank you.
Awesome, no problem. Awesome. So that's where, again, we're not asking for multiple things. We're just asking for one. And especially when it comes to the lysosome and peroxisome, they do have multiple functions, right? Because they break down things. And there's a lot of things to break down. But it kind of breaks down along those lines, no pun intended, where we think about the lysosome as kind of bacteria eaters. And we think about but I would say breakdown of bacteria. And then the peroxisome, we think about that in terms of metabolism. But they both function, in these breaking down. So kills bacteria is okay. Yeah, the breakdown of bacteria. I would probably say the breakdown of bacteria because that's more specific. An enzyme is going to break down something. So I would stick with the term breakdown just to avoid any issues, right? Breakdown of bacteria. Okay. Yep. All right. Awesome, guys. That's great. Now let's do a couple more, and then we can come back if we have time. I know you'd be so excited to do that. What about centrosome with centrioles? It's not weird. Again, Sophia, if that's what you remember, just make sure you just tell me peroxisomes, not hydrogen peroxide, and then you associate the idea of how that breaks down, right? Yeah, there's nothing that's weird as long as you associate it correctly with its biological function, basically. I mean, I think of catabolism as, as cannibalism. Not that it is, right? But that's how I remember it. Breaking down, which is very weird. All right, so, yeah, so There you have it. Just don't talk about cannibalism when you talk about the breaking of bombs. All right. So everybody, Christmas candy, exactly, Christmas candy. So you guys all like attacked it, so I can't even see it anymore. But the centrosome with centrioles, you have one centrosome with two centrioles, and that's the Christmas candy here. And you're right. Somebody mentioned before that this is clearer. It is. These were taken. A wonderful student, photographer. She came in and she volunteered her time to take these beautiful pictures. These are all our models from school. So it was really great. Yes, Christmas candy. It looks like Christmas candy to me. So it's really nice in terms of the picture quality. All right, awesome. And then let's do one more. What about, oh, I didn't show you the Golgi apparatus. Whoops. The Golgi apparatus actually is, yeah, you guys are awesome. It's the red part there. It's so cute. That's so funny. Yeah, it's the red part. So actually, I'm going to do two to this so that you can see the difference. That's right. Now, who can tell me what the Golgi apparatus does? Who can tell me what the Golgi apparatus? Sorry, guys, I'm going to kick you guys off so we can see it. The red part here. Let's see. Packaging center modifies. Now, I need to hear proteins in there, right? I need to hear modifying proteins. Okay, guys. So you gotta when you're talking about packaging and modifying, we gotta add proteins because that's really, really critical. Okay, exactly. It modifies the protein. So where do we make the proteins in the body? In the cell. Where do we make the proteins in the cell? So Melinda, it's already synthesized. We're just tweaking the protein. Yes. So we make it at the ribosome. Specifically, we make it at the rough ER. Now, on the test, guess what I want you to write? Rough endoplasmic reticulum. Don't give me rough ER. You're going to be losing points there. Everything the way it's spelled out here. So the rough endoplasmic reticulum is where? Now, here, I think you can see that it's really nicely. I know, sorry, guys. No RER, rough endoplasmic reticulum. How do I know? Because it's on my list. All right, yeah. Exactly. So everybody looks like they've got a good handle on it. Let me erase all of you guys anyway and show this here like this here like this. These are rough endoplasmic reticulum, the little red ribosomes, right? And that makes proteins. Exactly. These make proteins, right? Now, here is the smooth endoplasmic reticulum. What do they make? You see how there's no red dots on there, right? No ribosomes. So you're going to make fats, right? Now, in this case, go with just making fats because that's kind of more. When we're talking detoxification, yes, because it has a couple functions, and you don't have to go with fats. But each of these have a couple functions, right? Well, not all of them, but most of them have a couple functions. The smooth endoplasmic reticulum makes fats. So that's going to be what is outlined here. A couple people have their pointer on it. So the smooth endoplasmic reticulum. Okay, all of this is in your textbooks. You're going to drop it into a cue cards, and you're just going to memorize it. Ribosomes are the ribosomal yeasts that are on the either rough ER or they are free ribosomes. Not that part. That's the mitochondria, and they are critical in making proteins. Right. So we need this subunit, these ribosomal subunits, in order to make proteins. Without it, we cannot get the right amino acid and put them together. The function of the centrioles as part of the centrosome is that when we have the mitosis, actually, can you give me a couple minutes and we're going to go into mitosis? We'll talk about it there. Exactly. In cell division. Yep. Awesome, guys. Sweet. Awesome. I love it. That's basically it. Okay. We'll come back to this in a bit. And so if we spell it wrong, it's completely wrong. Nope. It depends. So the basic idea, Darielle, is that if it's basically, it can be like either 0.25, 0.5, 0.75. So if you get one letter off, I'm going to take a point out, a little bit off, right? If you give me something that I cannot understand, that's completely wrong. So spelling does count, right? If you give me something I can't understand, there's no points. If it's just a couple of letters off, there's a little bit of points off. So yeah, it does work that way. That's why you got to write it out. You got to write this out. Yep, I know. It is what it is, but that's just kind of the way it is. We've always done it this way, and I don't think there's ever been a discussion to change it. So, all right, good job, guys. So let's go ahead. We'll come back to this in a little bit. And here's, this is a second image that you'll find on rough ER synthesizes proteins. Yes, ma'am. That's exactly what it does. Yeah, makes proteins, synthesizes proteins. They're both the same thing. Okay. So, oh, by the way, guys, I have a little bit of a headache. So if I look like I'm bothered by you, I'm not. I'm just working through it. So don't worry, I'm fine. But I don't want you to think like you're bothering me. It's just I'm working through a headache. All right. Don't worry about it, honestly. I just didn't want you guys to think that I was like, oh, you're asking me another question. That's not the case. So don't worry about it. All right. So this is the second image that you will find on Blackboard. And so you're able to print this out. But I would work off of this one first and then go to this one, right? Just so that you're using this as kind of like a backup. Oh man, I don't even, honestly, I can't even, this is just a little headache. I just can't imagine migraines. Like this is literally, it's just it's just working its way. So I'm totally fine. All right, guys, I just want to let you know. All right, so where are we now? So again, notice what I did here. I just want to clarify something. So notice that for exercise 4, I didn't really go into the lab manual. Read the lab manual, right? I want you to. And it's great. You should read it. It's just, I'm making the decision as your instructor that for this particular part, I'm going to focus on getting ready for the midterm. Okay? So that's just kind of where we are with that. All right, everybody does things differently. Like if you go into Dr. Cedri's class, her class is going to be different. If you go into anybody else's class, because I don't remember everybody now. If you go into another class, it's going to be different. What date is the midterm for this class? Same window as the midterm. So you can go ahead and check your portion. You can go ahead and check your calendar. It's all on there, and it should be a similar window as well. So we have the same basic idea of opening it up and then closing it a little bit later. The windows are pretty much the same. But that's all on, you can download it on Blackboard, which I recommend you do in March, I think. Yeah, I think so. I don't have it all memorized, so, we'll just kind of keep it at that. All right, exercise 5. Again, exercise 5, we're going to be looking at mitosis, okay? Now, what we're going to look at here is just going through, for me, I'm going to go through the mitosis, right? I'm going to go through all of the concepts, and then we're going to go through the slides, okay? Oh, and the model. So we're going to go through the model and the slides. But first, I'm going to go through actually what is mitosis. That's later, but thanks. Yeah, awesome. So what we're going to do is we're going to go over what mitosis is, just a general idea. And then we're going to go over the model, because if you look on page 31, you can see we have something called mitosis models. And the pictures I have, because I have the correct PowerPoint, are going to be the ones that I got off of Blackboard, right? Now, I do often get an image that's already labeled because that's super easy, and then that helps you, and then we go back and forth. That's just the way I prefer to do things. All right, so mitosis models, slides, because you guys have, as somebody mentioned earlier, you guys will have histology assignments. So we're going to talk about that. Okay, today's class will prep you for that. All right, so let's jump in. So mitosis is basically one part of something called the cell cycle. The cell cycle is this entire circle here where we start at the growth of in this awesome little circle here. We start at the growth of everything you need in a cell, right? We're going to make proteins, we're going to make organelles, we're going to start to develop everything inside of a cell so that we can ultimately replicate that cell or duplicate it or make a clone, because we're talking about mitosis. I'm making the identical cell from the parent cell, right? We're not talking about germ cells or sex cells, right? We're not talking about sperm and eggs. We're not making an organism. I'm simply making a liver cell. I'm simply making a blood cell, or I'm making a cheek cell. I'm making any of the myriad of cells in the body by cloning it. Okay, so this is the process by which we make an identical cell. I'm going to get one identical cell from this process. All right, so a couple of things that are important. Mitosis is simply one piece of this pie, right? The vast majority of the cell cycle has to do with spending time growing things, organelles, etc., and then being in a phase called interphase. And this is important. This you may be asked on your lecture test. The DNA replicates in this phase between G1 and G2, shockingly called interphase, between phases. Just curious, how long is mitosis? Oh, hours. Hours. Yep, It goes in hours. I don't know. I'd like 6 to 8 hours for G1, I think, and G2, but I have to do a quick Google search. So G1 and G2 are going to be the areas where you can see here we're growing things. Now, why do we do that? We do that because we want to make an identical clone. And every cell that has all the organelles we just talked about, right? You've got to make mitochondria. You've got to make Golgi apparatus. You've got to make lysosomes, peroxisomes. You've got to make a bunch of cytosol and cytoplasm. So all of that stuff that we need, we have to make it, right? So all of that gets made. So this is pretty energetically intense, right? In terms of going from one cell to duplicating everything in that cell so I can get a second cell, okay? So the important part, in addition to what I just said, is that interphase is an important part because DNA replicates. So when I say DNA replicates, I mean the entire DNA sequence is going to be replicated identically. Right? And when I do that, I get, at the end of this process, at the end of mitosis, I get two separate cells, which we call daughter cells, and they are genetically identical. When we do meiosis, that's not the case, right? They're different because they are going to, they're going to switch a little bit of genetic material, so I've got some diversity, right? You don't want to look like a clone of your mother or father. That's weird. So you want to have a little bit of differences, right? And that's why we don't marry in our close family thing, because it's, you know, you can inherit a bunch of diseases, you know, kind of like the royal family throughout history. So anyway, that's neither here nor there. Just know that we're talking about a cycle where we're going to duplicate an entire cell, and part of that is to replicate an entire DNA sequence, or also called the genome. Okay, so that's going to be important. As we go into G2, we're going to be continuing with that growth, and we're going to prepare for cell division. Okay, so we've kind of made everything as we go into G2. And then here, when we go into mitosis, that's what we predominantly talk about. So it's really about mitosis, and don't forget interphase. Kind of the take-home message. We don't really go into that much detail with G1 and G2 because this is not a cellular biology class, right? We just want to understand, how do I get from one cell to two for somatic cells. Somatic means non-sex cells. Every other cell in my body other than my sperm and egg, right? So as I get to mitosis, you guys are probably familiar with the idea of PMAT, right? So this is where we have mitosis proper. We have prophase, metaphase, anaphase, and telophase. Okay, we do have cytokinesis, but you see here that we consider that basically the physical process of separating these cells into two. G1 takes the longest, right? I think it does because we're doing a lot of duplicating of stuff, right? So I got to make my Golgi apparatus and all the other stuff. I'd have to check, but they're actually, I don't think they're massively different in time, G1 and G2. I'd have to do a quick Google search. All right, so with cytokinesis, it's not technically considered a step within mitosis, which is that PMAT, prophase, metaphase, anaphase, telophase. It just simply means that the cytokinesis is the actual physical separation of now two complete cells. And then I'm just going to get my budding or my cleavage furrow, and then I'm going to separate them, okay? So when we get to the models in a couple minutes, we're going to see that we have an interphase model. We have a prophase, metaphase, telophase, metaphase, anaphase, and then telophase model, okay? We have a couple more models because technically the phases are actually early prophase, late prophase. So you're going to see that there are a couple different model images that could be considered prophase, right? But we don't go into the differentiation. Like, you don't have to know prophase 1 or prophase 2. You just have to know what's on our cell, our list, right? And when it comes to the mitosis model, which is what we're going to be looking at, you have to know interphase, prophase, metaphase, anaphase, telophase as the phases. How do I know that? Page 31. Okay, so we're going to go into the idea of what is mitosis, which is going to help you in lecture, and then we're going to go over the models, which is going to help you in lab, and then we're going to go over the slides, because you also have to know that. That's on page 32, and you have to understand that as you go into your first histology. So we're going to kind of all finish that up by 8:20. All right, so that's it. So again, this is all just a repetition of what I just mentioned. Interphase is not part of mitosis, it's part of the cell cycle, okay? There's a non-dividing period. In other words, this is where things start growing, right? We're not actually dividing. We're not separating chromosomes yet, okay? So again, the big thing that you need to know is that S phase or interphase, right? We're going to be replicating the DNA. So that's kind of your big take-home idea here. Okay, awesome. Now here, this is I really like the slide. So if you want to take a screen grab or a screenshot of this, you can. I like it because it breaks down each step as we're going from DNA and condensing it. So the big idea is once I've made all my organelles and I'm ready to physically separate into two different cells, one thing I have to do is I have to take that DNA that I've duplicated or replicated that I've replicated in interphase, and I need to condense. Like DNA is quite long. It's small, right? But it's really long, and I need to condense it into a form where I can actually separate it, and then have half go into my cell that I started with, and half that's genetically identical go into my new cell. So the idea here is as we're going through some of these phases, specifically interphase to prophase, we want to be subsequently getting smaller and smaller and more condensed DNA. And so that's what happens in this slide. And this is where we start adding more of these C words, right? Chromatin, chromatid, chromosome, right? And then we already have the centrosome and centriole. So you got to watch out your terminology here. And this stuff is going to be on the lecture, okay? So what do we have here? DNA, of course, you know the DNA has to do with a double helix, right? We're going to have a sequence of nucleotides, which are either A, C, G, T. And of course, AT are going to match AT, and CG are going to match, right? That's your nucleotide-based pairing. That's what we have in DNA. RNA, we have uracil instead of the thymine, okay? And so that's kind of what we're looking at here. Again, the point is that we want to condense all this DNA because we still need to take the duplicated DNA, the replicated DNA, and separate it. Apple car. Apple car. Apple car. Sorry. I don't understand.
To remember.
It's okay. I thought so, but
That's right. Apple car.
Oh, okay.
Apple tree, and then the other one is under the tree, and it's still the
okay.
You guys know what I mean.
I didn't with the apple car. I'll be sorry. I appreciate it. Thank you. Oh, you guys are seriously gonna kill me. I appreciate it, guys. Thank you. Oh wow. That was good. I appreciate it. It's just I am very easily confused. Not sure if you've noticed that. So all right, guys. You guys are good. I appreciate you. That was good. I like the apple tree because the apple tree is the adenine and then the thymine, right? The AT, and then guanine cytosine, GC. Uracil has to do with the RNA. So actually, there's no uracil in this picture. There's no U part of this because we're actually just taking the DNA sequence and we're condensing it down. All right, awesome. So I appreciate that. That was actually very helpful as well. And so what happens here is that as we're condensing, right, and I keep repeating the idea of condensing because it's actually going to make your understanding of the slides much easier, and it's going to make your understanding of the models much easier, which is, you know, my job, right? So as we go from the DNA, which is that kind of helix sequence that we see, like the twisted ladder, right? We want to actually continue to condense that. So the next step in the condensing process, or making it smaller and smaller, is making something called chromatin. So the way that I think about chromatin is this. Everybody is probably familiar with a spool of thread, right? If you have a spool of thread, it's like this wooden or plastic part that we wrap around the thread. We wrap the thread around, right? So that's kind of a spool of thread. Well, that's kind of the analogy of what we see when we look at what's called the histone. The spool part is considered the protein, right? This histone protein. And then the thread would be the DNA, the sequence of DNA that we literally wrap around this protein. Why? Because we're starting to condense it, right? We're starting to manage this DNA and we're starting to make it smaller and smaller, but in an organized fashion. We're not just taking like a bunch of DNA and shoving it together, pushing it somewhere, right? It has to be organized. So that's kind of what we see in what's known as chromatin. In other words, these histones start to come closer together, or my spool of thread example. So I've got a bunch of these histones like this, and they come together. And that is called chromatin. Okay, it's a chromatin fiber. And so I've gone from DNA, and I'm condensing into what's called chromatin. And then the chromatin is going to continue to condense in what's called a chromosome. And you guys are all familiar with the chromosome, right? This is where we've got our X-shaped pattern here. A chromosome is tightly condensed chromatin. right? And so we're able to condense all of that DNA that we duplicated, right? That we replicated. I keep using that. Technically, they're the same, but stick with replicated because that's what everybody uses in terms of terminology. So we take that replicated DNA, we're going to condense it from chromatin into chromosomes. Looks like Gumpy. It does look like Gumpy. It looks like the rubber guy. That's right. So here we have another term that we're going to add. So now we've got this nicely condensed, packed chromosome, right? And again, here we've got a lot of C words, right? We're condensing. We go from DNA, which is D, to chromatin. And when we get to the chromosome, which is the X, we have sister chromatids, okay? So each of the half of a chromosome is called a chromatid. That's why they're called sister chromatids. A chromatid is identical DNA. And that becomes really important in mitosis because we actually are going to see that we separate these chromatids and they become the DNA of the new cell. Okay, so here's another picture of that. Tids. I don't want to go there. Come on, guys, it's Friday. No, sorry. Yeah, chromatids. Sorry.
No, I was asking, were you saying chromatid or chromatin? That's what I meant.
I know. Sorry. Okay. Let me go back. Oh, I can hardly wait to read your feedback after the class. Okay. Exactly, Portia. You said it, not me. I didn't say it, okay? No, sorry, I think I lost my hearing there for a second. We're good. Oh, God, you guys are good. I'm going to answer your question because it was a good, it is a pair. Anyway, oh, God, you guys are horrible in the best way possible. Okay, so I'm going to answer the question. Yes. When we condense our DNA, we get chromatin. So yes, chromatin or chromatin. Okay. As we continue to condense our, wait, did I call it chromatin? Yeah, I did. Good, I'm right. Ooh, I was like, great, I made a mistake. So do you notice here that chromatin? Now I see that it says here chromatid. We'll go on to the next one. So I'm right. So here we go from a full DNA sequence to wrapping around the histone, the DNA around the histone, which becomes chromatin. So that's our tin. Okay. Then we take our chromatin or chromatin and we continue to condense it into the form of a chromosome. Now, half of the chromosome is the chromatid. So chromatid for the chromosome, half of it. Chromatin for the small parts of the DNA that are condensed. Is chromatin when it is not dividing. The division has already ended. So we finished the dividing in interphase. Once we finish dividing, we get this whole mess of, after we finish dividing, we get this whole mess of chromatin and then as we go into prophase, we start to condense into a chromosome. So the chromatin goes, it goes from the interphase into the prophase, and then we start to condense into a chromosome, which is made up of, and this was the question, which became very funny very fast. We go from a chromatin, which is still there. The chromatin is still there. It's condensed into the chromosome. And then we have two chromatids. One half of the chromosome is called a chromatid. The other half is called the sister chromatid or chromatid. Loose DNA, once we have a chromosome, we don't have any loose DNA anymore. And the chromatin is taking all that loose DNA and wrapping it around the protein called the histone. That makes sense? And if you have to remember, I'm not going to say it. All right. Yeah. Good. Good questions, guys. And yeah, sorry, it's Friday night. Gloria, you have a question.
So you would have twice as many chromatid as chromosomes.
Exactly. Exactly. So if I have 46 chromosomes, which I do, I have 92 chromatids. That's it. Perfect. And we're going to get into that. So I'm taking it really slow here because of all the C's. And you can see that we've gone from like chromatin, and now we're talking about chromatid. And it gets confusing. We haven't lost the DNA. We haven't lost the chromatin. The DNA has been condensed into chromatin, and that chromatin has been condensed into a chromosome. And the chromosome has two halves. Two halves, and they're chromosomes. I don't know what I did. All right, there we go. So that chromosome is going to have two halves. One half is a chromatid, the other half is another chromatid, and they're identical. I'm so immature, I can't stop. I know, Terilyn. It's because you're so immature. You opened the door. I felt like I had to walk through it. No, you're not immature. I know. It was funny. That's why I laughed. All right, guys. Back to biology. Stop your dirty minds. You're all dirty-minded people. All right. Wait till we get to the sheep. Wait till we get to the sheep brain and the corpora quadrigemina looks like a little butt, but that's a different story. All right. So, all right. So here it's just a second picture of. It's just another way to look at the chromatid. It's a little bit here. You can see that they've kind of outlined that chromatid. All right, now, centromere. I'm going to add the centromere. Centromere is just like a belt that holds the chromosome together. So for every chromosome, I have one centromere. And this is answering questions also from your little blue box on page 30. So what have we established so far? You guys can feel free to open your manuals to page 30. We have, for question 4, it asks about what's going on at the metaphase during mitosis when the chromosomes are lined up at the metaphase plate. At that particular time, as per this question, there's 2 centrosomes, right? Which means there's 2 centrosomes or 4 centrioles, right? Chromosomes, 46 in a normal somatic cell. Centromeres is going to be 46. If I have 46 chromosomes, I'm going to have 46 centromeres. Centrioles are 4 because for every centrosome, there's 2 centrioles. Okay, it's okay, Gloria. We'll get there. And then chromatids. Remember, for every chromosome, there are 2 chromatids. So 46 centromeres. Yes, 46 centromeres. Hold on, guys. I got to spell that correct because centrosome is different. Let me write all our C stuff. Are you guys ready? So, DNA condenses into something called chromatin. So, I'm going to separate my C's so that you understand. Chromatin, that goes to chromosome, which is made up of 2 chromatids. All right, you guys following that? DNA condenses to chromatin, condenses to a chromosome, which is made up of 2 chromatids. Now, we're going to take our next step. Centrosome is made up of 2 centrioles. Remember the little candies? The little Christmas candies on the cell model? Those are 2 centrioles. 1 centrosome is made up of 2 centrioles. So if we have 2 centrosomes, we have 4 centrioles. Everybody following me so far, guys? You can look on page 30, question 4. Hey, guys? Good. If there are 46 chromosomes, if there are 46 chromosomes, then I have 46 centromeres. There's no such thing as a centrosome. There is such thing as centrosome, but a centromere is separate. Centromere. I got to make sure I'm spelling it right. All right, 46 chromosomes come with 46 centromeres because they are like the belt of the chromosome. They keep the chromatids together. Okay, guys? And then, yes, and then for the last one, which is chromatids, there are 92 because there are 2 chromatids. For 1 chromosome, there are 2 chromatids. That's the picture here. Chromatids. I'm looking to make sure I'm spelling it because my brain's starting to fizzle out. All right, guys, make sense? One human, two legs, but I have no idea. I trust you, but I don't know. All right, guys. Does that make sense? Yeah, like a belt. Yes, it's a good analogy. It's just my brain is. All right, guys. Shannon, did that help? And I just want to make sure that somebody said that they were confused back here. Gloria, is that good? Are you with me? Hold on. Sweet. Okay, awesome. Yeah, it's been a long week. It has. And this is a lot of information. How many centrioles? For every centrosome, there are 2 centrioles. Now, at this point of the given example, where the chromosomes are in metaphase of mitosis, we have set 2 centrosomes, which means we have 4 centrioles. So write this down and think about how I would take screenshots of these pictures if I were you, so that I can separate them. All right, guys. So it's a lot, but by the time we get to the models, you guys will be able to tell me. And then most of it you'll be able to understand by the time we get to the slide. Yes, ma'am. Because for every chromosome, how many chromatids do we have, guys? You got it. And here it is.
Wait, excuse me.
Go for it.
Where's the centrioles? You're not showing that. I can see chromatids. I don't see that part.
Because remember, we talked about that as part of the model. So here is the centrosome. And for each, so a centrosome is consisting of 2 centrioles. So this is actually in the model, not part of the chromosome. That makes sense? Yep, that's why it's a little confusing. Because it's part of the cell, and we're going to see a picture of what that looks like when we get to the thing. And that's what I'm saying, because we have so many C words, it starts to jumble up together. Yep, 2 makes up a whole centrosome. Exactly, that's right. And that's why I kept calling it see how it looks like Christmas candy here? That's 1 centrosome made up of 2 centrioles. Now, when the cell is dividing in mitosis, and the chromosomes have gone to the metaphase plate, which I'll show you in a second, there are 2 centrosomes and 4 centrioles. And I'm going to show you a picture of that. In a non-dividing cell, there's only 1 centrosome with 2 centrioles. Does that make sense, Halima? Are we good? It's okay. I mean, it's probably just kind of okay now, because you might have to go back and just take a look at it. Okay, now I'm going to go into PMAT. So I just kind of briefly talked about PMAT when I talked about the piece of the pie, which is mitosis. And it's taken a little while, but it's just because some of these terms, as you can see, they're really easy to get confused. So it's better that we hash it out now. You can memorize it, but I find that sometimes it can get confusing, right? So we've answered question 4, by the way, on page 30. Now, keep in mind, I don't have to ask about 46 chromosomes. I might ask you a question like on your homework about 40 chromosomes, right? Just like the homework, if I have 40 chromosomes, then I'm going to have 80 chromatids, right? I'm going to have 40 centromeres, because that's the belt, 1 centromere for every chromosome. Okay, guys, that makes sense? So keep in mind, 46 chromosomes is what you're going to get in a somatic cell. I'm going to show you that in a second. But I could ask a question that's not typical, which, again, you saw in your homework. All right, guys, awesome, sweet. Now, here's a picture, just an illustration, typical illustration we see for mitosis. Here we can see interphase, and this is actually a really good picture because interphase has something very special, and you'll see it in the model as well. Interphase, you still have that nuclear membrane, also called the nuclear envelope, but it's called the nuclear membrane. In other words, we still have all of this DNA, which is starting to it's already been duplicated or it's replicated here in interphase, but we still want to keep that DNA together. So we're going to keep that nuclear membrane there intact. You can see that nuclear membrane under the slide microscope, usually. And when you see that nuclear membrane, that helps you to distinguish between interphase and prophase. So that's like a tip, right? And when we get to the model, you're going to see that in interphase, again, that nuclear membrane is intact, where as we go through prophase, that breaks down because want to obviously separate my newly condensed chromosomes. So that's a tip. That's actually an important thing. It saves you a lot of time when you're trying to differentiate between a cell that's in interphase and a cell that's in prophase. You can see the chromatin in prophase. Yes, but you can really see it here. Do you see this picture here? All of that squiggly lines, purple, that's chromatin. So you can see some chromatin, and actually you can see some chromatin, but it's really going to be in interphase. Because what happens in prophase, the hallmark of prophase is that you have the actual definition of prophase is the condensing of chromatin into chromosomes. And I'm going to show you that in the model, right? So you can't see as much chromatin in prophase as you can in interphase. And in later prophase, you can't see any chromatin. All you see is the chromosomes. Tanya, does that answer your question? And again, we're going to see that in both, especially the model.
So it gets darker in the nucleus in the prophase.
It does. It does. And it gets smaller. And the big thing is because sometimes it's a little confusing because there's going to be some pictures and you're going to look at it and you're going to say, I swear that picture is interphase, right? So remember, they're phases. That's why they're, so it could be like the end of one phase and the beginning of the other. The big thing is as squiggly and light and a nuclear membrane, right, around the nucleus where the DNA is, that's going to show you interphase. It still maintains that nuclear envelope. It's not as dark and it's very squiggly, which sounds super non-scientific, but you're going to see what I mean. Does that make sense? And then the prophase is going to get more darker, smaller, because you can't, under the microscope, you can't actually make out the chromosomes. You can make out where they are as we go through, but you can't tell an individual chromosome in prophase under the microscope. And I'm going to show you what that means. All right, so good questions, guys. And again, I'm going to expand on this when we get to the model and when we get to the thing. It's just like you see. So my method is let's get it and then let's apply it. Awesome. Thanks, guys. All right, so prophase here, we're literally going to be condensing all of this chromatin into chromosomes. That means it gets smaller, which is what we want, right? We want to condense it. A condensed process, think about condensed milk. If you've ever used condensed milk to cook a pie, I don't know why I thought about that, because it's condensed milk. If you got that little can of condensed milk, it's like thick. Exactly, it's thick. So you've taken something that you will add water to, and it'll become more like what you know is kind of milk. So condensed anything is a lot smaller, and we do that so that we can move things around usually, just like we condense the DNA here. So those chromosomes take up much less space. We still have all that DNA, but it takes up much less space in the form of a chromosome. So then we go into metaphase, which is so easy, and I almost don't want to talk about it. It literally, all my chromosomes line up in the metaphase plate, and it's very easy. And that's the scenario here that asked about question 4. So be careful, guys. All of the answers on question 4, they apply when we are in metaphase of mitosis. And it's because of this, what you can see here. Okay, and we're going to see those 2 centrosomes and the 4 centrioles as well. All right, and then here, here you would find a centrosome here, you would find a centrosome here, and they will pull apart the chromosomes, which we know to be anaphase. Okay, and then here, telophase, well, we want to bring this DNA back to the poles, the opposite ends. We want to start to form a new nuclear envelope because we want a nucleus, right? We don't want this DNA just all over the place. And then we're going to form our nuclear envelope again, or nuclear membrane, and we're going to keep that DNA together. And then we go, which is called telophase, and then we're going to go into the last part, which is creating 2 daughter cells, or cytokinesis. All right, and that's it. So now we're going to see all of this, but I want you to take a minute here. We actually kind of already talked about it, but I really like this slide, so feel free to take I found this here, you can see, and I really love it because sometimes it gets a little confusing, as we talked about it earlier. Feel free to take a screenshot. Obviously, you can take a screenshot of anything. So here, in a somatic cell, right, which is not a egg or sperm cell, right? Those are germ cells or sex cells. We're just talking about somatic cells. They are what's called diploid. So the haploid cells are the germ cells. Here we've got prophase, metaphase, anaphase, telophase. So you can see what's happening in each of these phases based on this chart. So we start off with 46 chromosomes, right? That's what the DNA condenses into, right? We've got 46 chromosomes. We know that each chromosome has 2 chromatids, so that's going to be 92. And notice that we maintain that number throughout part of mitosis. We maintain 46. Metaphase, we align in the metaphase plate. We keep our 46, so our chromatids stay the same. Now, notice what happens here. In anaphase, we separate, and I'm going to get to your question, Portia. In anaphase, we separate the chromatids. So half go up here and half go down here. That's why all of a sudden they say we have 92 chromosomes, which really is kind of confusing because we've just separated the chromatids. And that's why we have 92 chromosomes while having 92 chromatids. Germ and sex interchangeable for testing purposes. Yes, they are. In my class. If you don't have me for lecture, but it should be fine. For your lecture teacher, I would double check. But it is the same. Germ cells and sex cells are the same. All right. So here again, in anaphase, what we've done is we separate the chromosome. In other words, we take one chromatid to the top, one chromatid to the bottom. Do we need to know all of these numbers for lecture? Yeah. You'll be asked on one of these. Yep. Just understand that one chromosome is made of 2 chromatids and 1 centromere. Okay, that's a given. If it's just a somatic cell, there's 46 chromosomes. So then you can double it. But they might ask you, like the homework, what if there's 20 chromosomes? Then you have to know that there would be 40 chromatids, right? And you have to watch if they're asking a question where you are. If you're in anaphase, you see what's happened here? I broke my chromosomes, separated them I'm moving them to the opposite poles. They call this 92 chromosomes still. Wouldn't it be better to say 0 chromatids for anaphase and metaphase because technically there's no chromatids, just chromosomes? No, there are chromatids. Whenever you have a chromosome, there are chromatids. And this is what it looks like. 1 chromatid, 2 chromatid. On every chromosome, there are 2 chromatids. And that the split pieces in anaphase are now considered full. So this is where it's confusing because they still call the split chromatids chromosomes. I mean, I wish they would just call them chromatids, but they still continue to call those split chromatids chromatones. Whoa. They continue to call those split chromatids chromosomes. Even though they're separated, I know, but that's the way it is. Yep. That's why you'll hear the idea of chromosomes. That's exactly what this is. So this is not wrong. In anaphase, they still say you have 92 chromosomes, but they're actually 92 chromatids. But they call them chromosomes, even though we've broken them. Yep, I know. It's the way it is. Yeah, I know. I'm with you. Alrighty. All right, so this is why I like this slide, because it's super, super helpful. And I literally, everything that I found, this has been the most helpful. So I cut and paste it. So at the end of mitosis, right, we've separated the cells. What happens? Well, then we go back to 46. Right, because we want to go back to what we had before, right? So instead of, we're going to start again. The bottom line is we start again. Just think about it as resetting, okay? And then here they just describe that. They literally just kind of describe what's happened in that whole idea. So you begin in exactly why? Because you make an absolute clone of what you started with, right? That's the difference between meiosis. In meiosis, you end up with different, it's not necessarily that you end up with different chromosomes, but you end up with different genetic material. Where here you end up with where you start, right? Because we're making a clone. All right, so this chart is explained really nicely there. Okay, so take a screen grab. If you want, you can go here and read more about it, but I took most of the important stuff. But notice that I'm referencing it, right? I didn't plagiarize. All right, awesome. Okay, now let's get into this so that we can do our slides. Okay, but we've done most of the heavy lifting, right? So what do you think this phase is based on what you see here? So you can see a pretty distinctive, you can see a pretty distinctive nuclear envelope here, right? Or membrane. Interphase. This stuff here is called what? What is this, guys? An interphase. What do you think that? Exactly. Awesome. I love it. Chromatin. That's chromatin. Now, what do you guys think this is? Because if I can have you guys to please turn to page 31. And under mitosis models, they ask for the phases, so this would be called interphase. They also ask for centrosome, centriole, Golgi apparatus, chromatin, chromosomes, nuclear membrane. Okay, that's called the Golgi apparatus. Exactly. Boom, you're done. Twofer. You knew it on the other model. You know it on this model. Great. Exactly. That's what I want. Good job. Feel free to jump in, circle, grab your pointer if you like it. It's no problem. All right. Awesome. Now, this is interphase because, again, the big thing for me is this. You got your nuclear membrane that's still intact, which you can see here. And you've got your chromatin. There's no chromosomes here. I see no chromosomes. Now, please circle my centrosome. At this point, I only have one centrosome, but I have 2 centrioles. Yes. Awesome. That's what I want. Careful. Don't call a mitochondria a century. You guys are funny. Good. Jensen, you have a question. Hop on if you want to do your thing.
So on page 28, would number 1 be interphase?
Okay. Yes, ma'am. You're looking at 5 slides, correct? And you're looking at the top left, correct?
Yes, ma'am.
Yes, that's great. Actually, that's great. Everybody turn to page 28. This is really beautiful because it's very clear. So, Jensen, I appreciate that. Top left, you're looking at 5 pictures of slides. Top left, as Jensen mentioned, is going to be the interphase. It's bigger nucleus. That's the nucleus, right? You can still see a little outline around, which is great. And what you want to do immediately is go to the right. Because right off the bat, you're like, oh, I see it. Tell me what the right is. If the left is interphase, what's the right, guys? If the left on page 28 is interphase, exactly. Because we've condensed it. So Tanya mentioned it before. So is the in prophase, is the nucleus going to be darker? It is. Because we condensed it, so it's going to actually take up more dye, which is the point of these stains, right? And then you can see that it's smaller. And that should be the case if I'm condensing, right? Just like condensed milk. So that's perfect. So I appreciate that very much. That's exactly right. And then the rest is quite straightforward, right? You're going to have metaphase, second row left, then you're going to have anaphase and telophase. So this is a great picture to compare and contrast. It's very, very good. And when in doubt, no, that's good enough. Sorry. All right, that's awesome, guys. I love it. So where did I get this? I got this from online under the Blackboard. Print it out, label it. You're good to go. Okay, get your points. Again, where am I looking at this for what you need to know? Page 31 under mitosis models. So Jensen, I really appreciate that. That was great. Super helpful. All right, awesome, guys. Good job. Now, we have 3 pictures here. They're all of different stages of one phase. What phase is this? So they're all the same phase. They're just different. They're either early or late. Not yet. They're all prophase. You're going to see metaphase in a second. Don't let it stress you out. First of all, Gloria, go back to 28 for a second. Compare the top, the second to the right image on page 28. So there's 3 layers of images. The first layer has interphase to the left, prophase to the right. The next layer underneath that, the next 2 images on page 28, it's going to have anaphase, metaphase to the left, and then anaphase to the right. So that you can take a look at right now as we're going through this. So yes, prophase. How do you know this is prophase, first of all? You can see the size, the models. I see what you're saying, Gloria. Now, first of all, I want you guys to notice this. So this is good. This is why we do this, right? Exactly, early and late. Now, why this is... So first of all, you guys can tell very clearly that this is prophase, right? X, 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 X. Right, guys? Even here, X chromosome, right? X, X. So these 2, I hope you guys can tell prophase. Condensing to chromosomes. Exactly, Melinda. So here, I still have that condensing, right? Now, I'm just making it work here, though. This is very early prophase. But I want you guys to compare something. Look at interphase. Look at this compared to this. This is how you have to study these guys. If they look at all similar, you got to study them together. These ones are clearly prophase. I hope you guys can see the X's, chromosomes. Here, you might not be able to see it, but if you compare A with this interphase, they're completely different. No splitting. Is that here? There's no chromosomes at all. The condensing is just a very everywhere, right? Also, the nucleus starts to disappear. Exactly, the nuclear membrane. Exactly, and you can see that, which is great. You can see how it's really progressing through there, right? Exactly. So that's going to help as well. Somebody mentioned that was very difficult. It was very different. Hold on, let me see. The chromosomes are not lined up. No, not yet, because we're not in anaphase, in metaphase yet, guys. This is just prophase. Now, hold on. Gloria, are you good with the description and comparing interphase here with this beginning prophase? Awesome. Great job, guys. That's why we do this, right? Sophia, go for it. If you want to get on. Yep.
Yes, I just have a quick question.
Go for it.
What are asters?
Aster rays are these little things that look like kind of like angelic little things here, coming out here. Do you see how they're coming out of the centrosome? See these little things here? Those are aster rays, and they're not they're not the spindles down here. Notice how we're going through a progression, right? We're starting to form into something. Now here you can really see the aster rays. See how they're like this? It's like a little crown almost by the time we get here.
Got you. Thank you so much.
You are welcome. Absolutely, guys. So this is why we do this, guys, right? Because and in lab, I don't go into this much detail. I'm like, get a model, figure it out. And then I'll give like feedback. I am notorious for that in lab. I'm like all about histology because that's tough. I know it's true. I mean, I'm dead serious. I do that. So this is kind of special, right? Like I'm literally going through every list. In lab, I don't. I help you guys out in lab. But in this, I'm like, I will literally go through every list. Because what else are we going to do, right? Like, you know what I mean? So in that way, it's not as nice as on campus, but it's super clean. Like this is super clean. All right. Does that make sense, guys? I think we need to label the asters and the histology. Yes. The problem is you kind of got to like make up where they're going to be, right? So basically the idea is this. The take-home message for the aster rays or asters, depending on, they're generally called aster rays, but if they call them asters, that's fine. Notice where they are. Basically, your centrosomes are going to be at the opposite ends because they create what's called the mitotic spindle, right? Now, we want the term mitotic spindle because, well, how do I know that? Because if I go to page 31, I have something called mitotic spindle. Yes, it's the same as the spindle fiber, but we call it the mitotic spindle. So the aster rays are going to be up at the top of the centrosome, almost giving it like a crown, right? And that's what we see. So spindle fibers are microtubules that connect the chromosomes. Astral rays are the ones that mix. Yeah, I guess that's exactly right. So from our functional perspective, it's basically the astral rays are on the top. They're coming out of from the top of the poles at the centrosomes. Here, we're going to cross across here for the mitotic spindles in order to pull the chromatids when we get to the anaphase. Melinda, did that answer your question? Basically, yes. What you're saying is right. And you're right, Sophia. You do have to. But it's some of the pictures don't exactly show them. So you're going to point to where they would be. All right. So let's keep going. Now, what's this, guys? I remember from last month. You're right. All right. Anyway, metaphase. Exactly. Good job. This is very, very straightforward. It looks exactly like that. Now, here we've lined up our chromosomes, right, in the middle. We haven't separated them we haven't kind of separated that chromatid. But here we've got here we've got one centrosome with two centrioles. Here we've got a second centrosome. And then we've got the asters at the top coming out, asters at the bottom. And here you can see, again, it's the same idea of that mitotic spindle. Right? Call it mitotic spindle, even though spindle fiber is correct. Yes, exactly. Awesome. That's it. Woohoo. You guys are great. So now, this is where we don't want to get complicated, but just notice the difference of things, because our model is super, like here we have early and late stages of stuff. So actually it's not metaphase at all. Metaphase is the only one that's super easy. Metaphase is always in the middle, nothing else. Exactly. Notice this, even though it's close, we're still separating. The only thing you're going to get for metaphase is together in the middle. Yes, But remember, we don't ask for early or late anaphase. Just tell me anaphase and anaphase. Now, you might ask me, Miss Nicholas, that kind of looks like telophase, right? You got to compare, right? Obviously, this is anaphase. I think this is obviously anaphase because it's not in the middle, right? Ana apart, exactly. Perfect. Some of you might be like, wait, dude, that kind of looks like telophase. Well, let me show you telophase. There. You see that? We've gotten right into the poles. So we have separated to the end of the cell, right? Yes. Now that's called a cleavage furrow, and you can kind of see it here, but I think we're almost always going to give it to you here. It's just starting to bud, right? It pinches. So cytokinesis. Now, cytokinesis is actually the process by which we separate entirely. So yes, this is kind of the beginning of cytokinesis here. But I want you to notice something. On page 31, right, because we're getting you ready for the midterm. On page 31, we've got a couple things. We've got daughter cells. We've got telophase, daughter cells, and cleavage furrow. So cleavage furrow is the little budding part here, right? It pinches. We also have it here. But we don't have another model. Like this is the last model in the phases of mitosis. So you want to call this one daughter cells. Yes, I know. Cytokinesis has not completed fully, and a daughter cell is two separate cells. Unfortunately, they didn't give us that in the model. So this is what happens. Exactly. You completely separate the plasma membrane. You're absolutely correct. But unfortunately, we don't actually have one that shows two complete separate daughter cells. So if you see this one, call it a daughter cell. Okay? Because we're going to finish this cytokinesis here, and then you will get a daughter cell. And notice how we've reformed that nuclear membrane. You get the indentation, exactly. All right, guys, are we good with that? That's your model. And you have gone over everything, right? So it's not that bad. And this is what you can think about it. We've gone over two models, but we've shared some of the material, right? Golgi apparatus on both of our models, centrosomes, both of our models, nuclear membrane, both of our models, metaphase in the middle, and telophase. Telophase is here. Watch out this late anaphase. Watch out this late anaphase with this telophase. See, we've gone right to the pole on that one. And this one, you can see we're starting to reform that nuclear membrane. Yes, awesome. That's great. All right, awesome. Good job. A is telophase. Yes, ma'am. A is telophase. B is what we're going to call the daughter cells. Remember, we don't actually have cytokinesis as a question. So, guys, study what's on the list. That makes sense, guys? Will there be early? No. Now, Darielle, very quickly, look on page 31. Look under mitosis model and just quickly look at that list. If it's not on that list, it's not on the test. Literally, that's exactly what it is. Now, yeah. Does that make sense, guys? Exactly. That's why if you're like, why is this woman going over this list? Like, what the heck is she doing? It's because that's the test. Don't spend time studying. That's why, guys, I love all my students, right? I love all my students, right? Just like you love all your children. You're not my children, But what happens is I've had people spend so much time going through the manual, studying stuff that's not on the test. I'm like, why? Why did you do this to yourself? Dude, getting ready for the lab midterm. That's what you focus on. If there's no cytokinesis, you don't try to find it. Right? If there is, you try to find it. The mitotic spindle is attached to the chromosomes and the centrioles. Yes. They're basically from the centrioles here, the centrosomes, right? The centrioles it's the same thing, really, but the centrosome is made up of two centrioles. And the chromosomes get pulled apart along this protein conveyor belt, if you will, right? Because we set up in metaphase, and then we're pulling along this conveyor belt known as the mitotic spindles. Page 32, identify the red and white blood cells. Page 32, identify the red and white blood cells. Yes. Let me think about that. At some point, hold on one second. I'll have to check that. I'll add it next time if we need to. Red and white blood cells are super, super easy. They're just literally every every single little thing that looks exactly the same are red blood cells, white are purple. That's it. That's like literally the difference. And you can see it on page, where is it? We have it in here somewhere. Sorry, guys. Spermatozoa. Did I miss that? Let's see. Did we cover spermatozoa? I don't think we did. Let me add it to next week, and I'll just add it as picture. I don't have it this week, but actually, you know what? I don't really have to, only because it's so easy. Page 23, guys. Literally, page 23, top of page 23, red cells are all the little red circles. Anything with a purple that's fat, big, and has purple in it, white blood cell. How did she lie? She doesn't lie. She's amazing. She cited the model that was most obvious. Which one? In which one? What do you mean? Gloria, which model do you mean?
In the lecture video, she's like, we'll never test you on a model that you can't really tell what phase is in. We'll use the most obvious one.
Which is true. So if we go by that, then this is what they're going to do. I think this is the most obvious anaphase. This is me. I think this is the most obvious because I don't want to confuse you like, oh, is it telophase, right? So this is absolutely not metaphase. So I would say this is the most obvious anaphase. I need to bring all of these because, you know what I mean? I just want you to compare it so you guys can see. Now, this is the most obvious telophase. There's absolutely no doubt about it, right? So metaphase, there's only one, right? So if we're going to go by that, this is the most obvious anaphase. But I feel like I have to tell you both because I feel like at least I give you that information. Does that make sense, Gloria? So I feel like I don't feel comfortable just giving you one because what happens if they decide to give you that one? You know what I mean? So I'm giving you these so that you guys have the tools you need. But she's not. We do want it to be the clearest one. So what is A? A is early prophase, right? And you can see very clearly the difference between A and this one. Interphase has just a bunch of chromatin. That makes sense, guys? Cleavage furrow close at the beginning is when? Close at the beginning. Okay, so technically we can get the cleavage furrow. You can see how it starts to form here in late anaphase, but really we're gonna kind of show it here in phase. Kind of like that, what Gloria said. Where's the easiest thing? What is A? A in this picture, Estella? A in this picture is telophase. That makes sense, guys? Portia, did I answer your question? I'm not quite sure. Yes, A is telophase, and that's most likely where we're going to ask about the cleavage furrow, because you can know it's so clear. I mean, theoretically, we could ask about it here, but we really want the daughter cell here because they don't give us a model with two actually separate cells. That makes sense, guys? All right. So the question also, and that's a good question, guys. A couple of, I looked at page 32. If in terms of the spermatozoa, and the red blood cells, if you look on page 23, the sperm literally looks like sperm. Like if you've ever seen like a movie of like a little tadpole, baby tadpole, that's what the sperm is. And then the red blood cells are there. It's on page 23. The big cell that has any purple in it is a white blood cell. That's it. Round, exactly. Most of the cells are red blood cells. Gloria, did you have a question? Do you want to hop on? Okay, cool. All right, guys, awesome. So we still have, we're doing good timing, and you're asking me questions that are good because I realized I didn't do those 3 slides with my other class, but again, they're like literally the most straightforward slides that we have. All right. So lessons online histology. So the information, sorry, I forgot to turn off the mic. Oh, no problem. All right, guys. So lessons online histology. So for your first histology homework, it's on mitosis, right? Awesome. So basically you go on the Blackboard, you go to lessons, you scroll down, and you go to online histology. Now, if you stay at the top of lessons, under your lab, obviously, there's actually a, Go to lessons and then scroll down to online histology. When you click on online histology, at the very top, there's a video. Watch the video. They tell you how to do everything, right? And then you're going to go down, you're going to scroll down, you're in Blackboard, and you're going to scroll down, and it'll say, first histology assignment, mitosis. After you watch the video, those directions will make much more sense, right? You literally want to follow the entire thing, how to label it. The take-home message is that when you see that first histology assignment, it's a link. The directions are right there, but you click on the link and it opens up a PowerPoint. You download that PowerPoint, and then you use PowerPoint. You use straight lines, horizontal straight lines, which they'll show you in the video. I'll show you in a little bit, just on a slide. And you're just going to label everything they ask for, right? Everything's going to have a title. This is, 1,000 times magnification. They tell you what to say. Title says this. This is the magnification. And then they're going to tell you everything you need to label on there. Okay? So some of this I go over and some of this I don't, and that's probably why you guys are asking about it. I just kind of go over some of the basics around interface, around the different phases, right? But all of it is everything that you're supposed to label is right there, like literally step by step. How you label it is you click on the link, open up the PowerPoint, download it, use your fancy PowerPoint skills to put a line in a text box, and then you're going to label it exactly the way it says. Then you're going to save it. I would save it as a PDF because it's just easier. And will we do something similar in the midterm too? No. Why we do this is because we want you to just study the stuff because you'll be asked about it. But you will not do anything like that on the midterm. You will get pictures of these images and you'll be asked questions about it. But the questions you will be asked are on page 31 and 32. Now, the only difference is this. Okay, I do want you to turn to another page, guys. In your lab manual, please, page 40. Okay. Now, remember, we haven't actually gotten into any of the hardcore cell stuff, right? We haven't talked about squamous epithelium. We haven't talked a little bit on page 23, but we haven't talked about any of the actual cell types, the tissue types, right? When we get into that, we're going to go into more detail about the labeling, right? In other words, what could we ask, right? And all the questions we can ask about the cells next week are on page 40, but we haven't gotten there yet. So I'm kind of trying to pace ourselves, right? I want to stick to what we need to know. Do we have lab drawings? So Samantha, that downloading of the PowerPoint and labeling it, that's the lab drawings. So you're not actually drawing anything, you're labeling pictures. Yeah. All right. Sorry. So yeah, so you're technically not drawing anything. You're making a digital atlas, a digital slide atlas. I know, they were good. The drawings were good. I mean, that's why I don't spend all my time on the histology, and definitely not all my time on the drawings, but almost 30% of your exam is going to be a slide. So, yeah, we want you to get 100%. I work with my students. All right, guys. So let's look at this, guys. You can't draw to save your life. Trust me, it's not about how well you draw. It's about how you can translate what you see under the microscope. That's really the skill. So guys, here, I got these from the internet. Woo, they were labeled. So I'm like, cool, I'm going to use these. So here you can see very clearly that we've got, what's that? Perfectly middle chromosomes. What is that I just circled? What phase? Metaphase. That's it. Like, metaphase is the easiest one. It's like, you got stuff in the middle, no problem, right? So what gets a little tricky is when we talk about other things. Now, this one is a great example of something else. This is perfect, like absolutely perfect. And so metaphase is my favorite too, because you can't go wrong. Anaphase, look at that, perfect anaphase, right? Like absolutely perfect, beautiful. Now, sometimes it gets a little confusing, like do we have two cells smushed together? Or do we have telophase, right? So that's a little bit tricky. Basically, when we do telophase, we're gonna, like something like this, we could consider, hey, maybe that's telophase, right? Look at them smushed together. Looks like they're still one, they're still together. And maybe we've got some movement of our nucleus, because telophase, we're creating that nuclear membrane, right? But again, whether it's perfect or not, it's okay, because we're not going to show you two things together unless it's telophase. Does that make sense, guys? If we're going to show you anaphase, it's going to be one cell. If we're going to show you metaphase, it's going to be one cell. If we're going to show you something that looks like they're stuck together, two cells, then that's going to be telophase. Boom, right? Because sometimes it's hard to tell. We're not going to worry about that. We're going to, like Gloria said, we want the clearest image that we can get for you guys. All right, awesome. Now, so like I like this cell here for interphase. Let me show you what I like. There's a ton of interphase here, let's be real. But you know, I kind of like this bad boy. Why? Because you can kind of see this dark part around it, and it kind of looks all chromatiny, right? All over the place. So that's what I'm looking at, and I'm like, oh, that kind of looks like interphase to me, right? And then we want to start condensing into something small. Like for me, look at this. I'm going to show you what I think is a beautiful looking. I mean, look at this. I cannot actually circle it. Wow. Here we go. That looks like a pretty nice prophase to me, right? Definitely not metaphase. There is no doubt that we do not have a straight line there, but we're really starting to condense that material. And you can see that there's a big difference between these two cells in terms of what we see over here and over here. So we really want there to be a difference between interphase and between prophase, right? We want to condense that material. So now remember, they're phases, right? So an early prophase may look like a late interphase, for example, but we don't want to do that to you. We want something that looks nice and big. You know, look at these big cells over here with these massive nuclei and the circular, and we can see that nuclear, just like that pink line around it. All right, was there a question? Are these split? I could circle. Okay, let me erase and then you circle. Go ahead. What do you mean, Gloria? You circle what you mean. Yeah, those ones are janky. I wouldn't even look at that. Right? Anything like that. Like, you know what I mean? Like, seriously, because they're phases, it's like, oh, man, that's janky. So we don't want to pick. So this goes back to what you mentioned. We really want to choose something that makes you say, that's a big nucleus. I can see a dark line around that. I'm going to go with interphase, right? We don't want to pick something where you're like, man, I don't know. And because they're phases, it could be the beginning of one, the end of another. Yep. Jensen, do you have a question? Come on. Oh, sorry. That seems kind of rude.
It's fine. It's fine. So, when you're thinking about it, I'm going to see if I can try to explain it because it seemed hard for me.
Yep.
When you're thinking about every single cell in your body, is every single cell going to be at a different phase, or they're all going to be the same?
I see what you're saying. So every single cell that's not a sex cell, in other words, not an egg and sperm, and obviously you don't have sperms and the boys don't have eggs, right? So these things have to unite. That's reproduction, which we talk about later. So what we're talking about here are any of the cells in our body that we need to replicate. Now, keep in mind that some cells replicate a lot and fast. Your skin's, right? They're going to replicate super fast. Why? Because we wash our hands all the time. And our cheek cells in our mouth, we eat, so we slough them off. So different cells in the body are going to replicate at different rates. Some of them all the time, right? Some of them at much less rates. But this applies to any of the cells in our body at any given time. So yeah, at any given time, we have cells that are in any of these processes. Did that answer your question?
Yes. Thank you.
You're welcome. And again, we're just kind of laying the groundwork. So it becomes a little overwhelming to consider that we're made of billions of cells. And at any given time, this process is happening with any of our organs, right? So we've got cells everywhere. We have red blood cells. We have white blood cells. We have cells that are in our liver. We're hepatocytes. Those are our liver cells. We've got cells in our stomach. We've got cells that make up our stomach. So we've got cells everywhere. Any organ system is made of a bunch of cells. And because they're not germ cells, right, they're not sex cells, we've got mitosis happening at different rates. And that's why the cell cycle is why, for example, so there are some cells also that don't undergo mitosis, guys, right? So the heart is you don't make more heart cells. So it's not unified across and our muscle cells, we don't make more muscle cells. We don't use mitosis for this. Our neurons, they don't undergo mitosis, right? So our liver cells do. That's why you could actually lose part of your liver and it will grow back. Now, it depends on how much, right? But our liver can grow back. Why? Because of mitosis. All right, so I hope that kind of made it a little bit easier. Atrophy. Muscles atrophy. I think that would be the only thing that atrophies. So basically, with a lack of use, your muscle will get smaller and smaller. But remember that atrophy means smaller. It doesn't mean less. Just like hypertrophy, HYPERTROPHY, is the opposite. You make bigger muscles, but you don't make more muscles. We're part starfish. You go with that. This is so wrong. All right, awesome. So we're starting to all fade into the night here. But this is kind of what I wanted to show you guys. All right, so we're doing good, right? This is awesome. Oh, I love this cell. Do they call it, see, they call this prophase. I personally think this looks like a beautiful anaphase. So ignore this guy. They suck. I personally think this looks more like prophase, right? We've condensed it. We made it smaller. To me, this looks like a beautiful interface, right? So again, right, it's kind of a little bit subjective. What we want to do is we want to give you something that's really clear. Look how big this is. We've got a lot of condensing. As we get smaller, something like this, even something like this, you can see we're still condensing, going smaller, smaller, smaller. Where we start to change that condensing is when we get right into the metaphase, and then we start to separate those. All right, awesome. And that's why we want you to do the assignment, right? Because then you can label it. See, let me see. Blue phase, interphase, yellow arrow. I don't know what this is. But to me, I see some really beautiful nuclear membranes. Yeah, isn't it a beautiful pic? Very pretty. Awesome. I like it. And to me, I'm like, that looks like a pretty solid pink line on the outside there, right? That looks pretty hardcore nuclear membrane to me. Okay, awesome. Yay. That's it. And then again, just some more really pretty pictures. And as was mentioned earlier, which I appreciate it, page 28, you've got that's a beautiful picture of interphase. Not interphase, it is, but prophase in your text, in your lab manual. Like really, really nice, really clear, distinguished. That's what we want for you guys, right? We don't want to be like, is this late phase? You know, is it late phase, is it late interphase, early prophase? Like, that's crazy. We don't want that. All right, guys. And this is telophase, right? So you can see what we're talking about. Sometimes it's actually kind of hard to tell because, hey, is it just two cells smushed together? But it doesn't matter because as long as they look like they're together and we're going to put an arrow, you're going to tell me it's telophase. Okay? And that's it. Now, yeah, and that's it. So again, for the mitosis, for the histology assignment, you're not drawing anything. Like, there's no physical drawing. We actually do physical drawings in the lab on campus. So you're going to go to Blackboard. You're going to go to lessons. You're going to online histology. Click, click, click. You're going to watch the video. Make life easy on yourself. Then you're going to go down. You're going to scroll down to first histology assignment, parentheses mitosis. All the directions are right there. Then you're going to click on the link and you're going to open up the PowerPoint. Okay? Download it and then answer all the questions. So very quickly, because we have a couple minutes and I'm literally done, I just want to look at something if I can get it fast. Oh, I'm going to go look on hold on. I just want to look at something. I want to look at the list. So if you're wondering what I'm doing, I'm going to lessons myself. So actually feel free to go to lessons on Blackboard